Laboratory safety. Precaution for working with disease agents should be used by every microbiological laboratory to prevent one or more of the following: accidental infection or cross-contamination within the lab, contamination of spread of infection to adjacent, spread of infection through released biologics, infection of the laboratory personnel. All microbiological laboratories have probably experienced cross-contamination either in the form of mixed culture of bacteria or unwanted or mixed infection of experimental animal tissue culture. Readjustment from such accidents are costly and time-consuming. Numerous examples of the escape of disease agents from the veterinary laboratory have been reported or privately admitted. This type of disaster threatens both animal and human health and can cause great economic loss. The worldwide literature contains reference to approximately 6,000 cases of accidental infection of laboratory workers, and undoubtedly thousands more were not reported. In studies of laboratory acquired infection, the percentage by types of agent were as follows: bacteria 49%, virus 31%, rickettsia 11%, fungi 5%, parasite 2%. The percentage for outcome of the infection were complete recovery 70, permanent disability 26, death 4. In supplementary study with 60%, that is, the fatality percentage for the type of agent were virus 7%, bacteria 4%, rickettsia 2%, fungi 2%, and parasite 0%. Among all these infections, about 20 of the cases may have been due to the unsuspected release of infection aerosols. Many of the laboratory acquired infections are due to zoonotic diseases. Zoonosis disease agent of animal transmissible to human. This infection may result from handling the disease agent in the laboratory or from contact with infected animal, their tissue or excretion. Latent infection with the B virus in Asiatic macaque and with Marburg virus in African green monkey render these animal and tissue culture prepared from these potential hazards. Inadequately safety test biological material and vaccine shipped or released from microbiological laboratory have been the source of infection in some instances. Foot and mouth disease infection have been traced to smallpox vaccine and pediatric extract. A release smallpox vaccine was contaminated with Newcastle disease virus during World War II. Many servicemen contracted hepatitis from infection serum used in conjunction with formulation of yellow fever vaccine, and more recently, there was a tragic episode for polio vaccine.